starting a new unit on genetics, and genetics is a bit of a lengthy unit for us. We're going to go through a total of 8 chapters, all combined. We're going to break it up into 2 sets of 4 chapters. Chapters 9 through 12 will basically be kind of an old-style look at genetics, kind of pre-1970. And then from chapters 13 through 16, we're going to look at more a modern form, what we sometimes call molecular genetics. Well, with our basic look at genetics here, we need to start at the most basic level. We can't understand genetics unless we understand how cells divide. So in chapters 9 and 10, we're going to be looking at the 2 types of cell divisions. In chapter 9, we're going to hit the basics here. And in our first video, chapter 9, part 1, we're going to really focus on the key concepts of cell division itself. So in cell division, we need to remember what's really happening, what the important things are. And we are only going to look at eukaryotic cells. And so we're looking at things like animal cells, plant cells, fungi, protists. And for any of these type of cells, when a cell divides, there are 2 things that the offspring cells, or what we usually call the daughter cells, have to receive. Each cell has to get a complete set of chromosomes, enough genetic information for that cell to be able to carry out any processes that it needs to. And also each of the daughter cells will need enough just cytoplasmic material, enough organelles to survive. They've got to have things like mitochondria. They've got to have enough internal membranes to be able to carry out the life functions. So those are 2 completely separate steps. We're going to look at how they divide their chromosomes, and then we'll look at how they divide their cytoplasm. Now, there are 2 different types of ways that the nucleus can divide. So this first step of getting a complete set of chromosomes can be done 2 different ways. Those 2 ways are mitosis and meiosis. And if we're going to be really particular here and watch our language and our vocabulary very carefully, mitosis and meiosis are terms just for the division of the nucleus itself. They're not terms for the cell division. So sometimes we'll talk about mitotic cell division and meiotic cell division, meaning cell division using one of these 2 methods to divide the nucleus. Now, realistically speaking, we sometimes get kind of sloppy and just refer to the overall process as mitosis, including the entire division of the cell. But the difference between the 2, between mitosis and mitosis we sometimes call normal cell division. It's used anytime you just need to make more cells, so something multicellular like us can grow, or if you want to replace some cells that need to be gotten rid of, some cells that are dying off, too old, or whatever, you can replace them with mitosis. So this is just the normal everyday way of making copies of cells. Meiosis serves one purpose in animals. It's only used to make gametes. We're going to talk more about how it's a little bit different in plants next semester. For now, let's just remember that meiosis is used only to make gametes. So those 2 terms are each terms for division of the nucleus, but then we also have division of the cytoplasm, which is called cytokinesis. And cytokinesis is going to follow either mitosis or meiosis for the overall division of the cell. Now, before we get into how these things take place, we need to kind of back up a little bit and make sure we all understand what chromosomes are like. So we're going to start with the structure here. And we need to remember that a chromosome is 1 molecule of DNA, but that molecule of DNA is extremely long and extremely thin, and therefore very, very fragile. So to keep it from breaking, there are proteins there. The proteins give it some strength. They give it support. They give it a way to kind of condense itself and not end up getting tangled or broken inside the cell. And before cells can divide, each chromosome in the cell must go through a process called replication, which means they make a copy of themselves. Now again, if we want to be really particular about vocabulary for a bit here, a chromosome technically is a structure that only is found during the process of cell division. In between those divisions, the chromosomes kind of uncoil into a less organized form called a chromatin. But for the purpose of making things easier right now, we'll just refer to the structure as a chromosome, no matter what state it's in. Now, after a chromosome replicates itself and now has a copy, there's a structure called a centromere that holds those two copies together. Those copies are sometimes called sister copies, and they should be completely identical, barring any mutations. They would have all the exact same sequence of A's, T's, C's, and G's in the DNA. And each side, each piece of this chromosome is referred to as a chromatid. We'll look at a diagram in just a second here. But the centromere is going to also be involved during the process of cell division as a place where microtubules can attach and can move those chromosomes around. So this is kind of an attachment point for the structures that will cause the chromosomes to move. Now here's the diagram of a chromosome, a very simple one, but it kind of shows you the blue lines are showing you how there is a sequence of DNA that is kind of all spiraled up here, and the proteins aren't being shown. All we're seeing are loops of DNA. Now the reality is, magnified at this level, you would certainly never see the strands of DNA. They'd be still way too thin to see, so this would be coiled up much, much more thickly than what's shown in the diagram here. The centromere is this red dot structure in the middle holding the two sides together. So one chromatid we would represent by looking at just one side here. If we want to highlight one side, here is one chromatid marked in yellow, and we'll mark the other side in purple. And those two sides end up each being exact copies of each other, and they are going to be genetic information that the cell needs. And inside the cell, there are going to be multiple chromosomes. And you can see on the side here, lengthwise, these things are usually between 0.2 of a micrometer and 20 micrometers. So something that we can see the structure underneath a light microscope, which tells you that DNA is in fact a very, very large molecule. Now, a key concept in cell division is the idea of chromosome number. And that's one of the things we're going to be looking at the most here. And as we're talking about chromosome number, we decide it's helpful to divide the cells up in the body into two different types. So some of the cells in the body we call sex cells, otherwise known as gametes, that you probably remember. And the other cells, the ones that are not sex cells, are called somatic cells. Soma means body, so these just kind of mean general body cells. Any cell that's not a sperm or an egg cell. And this division between sex cells and somatic cells is a difference of chromosome number. All of your somatic cells have pairs of chromosomes in them. For every chromosome, there's a partner chromosome. And in mitosis, which we're going to be looking at here in Chapter 9, the whole concept of mitosis is to maintain that number of chromosomes in somatic cells and keep it the same from one cell to the next and make sure we end up with an exact set of all the chromosomes being passed along. Now, because we are humans, we tend to focus on humans the most, and the number of chromosomes in a normal human cell is 46. That number isn't special for any other species. There are other species that have 46. There's nothing that the number can tell you about a species. But just in terms of just memorizing the fact here, humans do have 46 chromosomes. That's something we do want to remember as we're talking about what's going on inside a cell. Now, gametes, those sex cells, have unpaired chromosomes. What happened is when the gamete was formed, only one chromosome from each pair in the original somatic cell that it was formed from was passed along. So if we're talking about a human cell, again, a human gamete, if it's normal, we'll have 23 chromosomes, half the normal amount. And if you pick any other species, if a species has something like 104 chromosomes in a somatic cell, then they'll tell you there'd be 52 in a gamete. It's always going to be half the normal amount. And so the chromosome number, like I said, is going to be crucial as we're doing cell division. This is a photograph of the chromosomes taken from one normal human cell. Now, they don't always line up like this perfectly inside the cell. This is something that was done with manipulation by a technician who took a photograph of the chromosomes and then arranged the pictures of the chromosomes to match them all up. And we can see in each pair, if you take something like pair number 8, we see the two chromosomes look virtually identical to each other. you can see on some of these, like pair number 3, we can see dark and light banding patterns, and those banding patterns are identical in the two. They're the same size, they're the same shape. The little indented areas, such as you see along each one of these chromosomes, that's where the centromeres are located. Centromeres sometimes are near the center of the chromosome, sometimes they're near one end, sometimes they're at an extreme end, like in pair number 14 here, they're way up at the top. And we see all these pairs of chromosomes until we get to the last pair, the 23rd pair, which we don't number, we just call it the sex chromosomes. May be a pair. You may have two that are both called X chromosomes, or they may be unpaired, like in this one where there's one X and one Y. And that tells us we're looking at a male, that these chromosomes were taken from a male human. Well, we're going to be looking at the cell cycle, which is a fancy name given to the overall process of cells dividing and then going through their normal processes and then dividing again and so on. And I want you to take a look at a quick little video. This is sped up, but this is a look inside a single cell going through the cell cycle. And we're going to focus just on the nucleus of it. And you can see the chromosomes showing here. And the chromosomes are being moved around by microtubules that aren't visible under this type of microscope. So it looks like the chromosomes are just kind of worming around on their own. But they actually are getting help being moved around by microtubules attaching to the centromeres. And we can see the chromosomes kind of lining up along the center. And after they're all organized, and it won't continue until they all get there, a couple stragglers kind of finding their way a little bit later than the others, once they all get there, then each chromosome splits apart from its sister chromatid, and they move to two new nuclei, and they split apart. What we're going to be doing in the second half of Chapter 9 is taking a look at that process in more detail and describing what's happening in each step along the way. And we'll go through the entire cell cycle in the second part of Chapter 9. We'll talk about the steps. We will talk about how these things are taking place and also have some reminders as far as what's happening when, all in the second half of Chapter 9. But right now, that's the end of the first part of Chapter 9. And as I keep saying, if you have questions, please bring them to class. Anything along the way here that you aren't understanding or want further explanation on, definitely bring it to class and ask those questions. But otherwise, we're all set, and you can have a good day.